Welcome to Brain Science, this podcast where we explore how discoveries in neuroscience are helping unravel the mystery of how our brain makes us human. I'm your host, Dr. Ginger Campbell, and this is episode 179. If you're curious about how your brain really works, this is the podcast for you. Today's episode is our 14th annual review episode, but before I get started, I want to remind you that you can find complete show notes and episode transcripts at brainsciencepodcast.com. Brain Science is produced independently and relies on the financial support of listeners like you. To learn more, please visit brainsciencepodcast.com/donations. Please send me feedback at brainsciencepodcast@gmail.com. You can get our free newsletter by texting the word Brain Science, all one word, to 55444. Brain Science to 55444. There's also a link to the newsletter in the show notes. I think we'll all agree that 2020 has been a very strange year. So I feel fortunate that I was able to release 14 episodes, including 12 entirely new ones, which is the most since 2008. Of the 14 guests that appeared this year, 7 were brand new, while Dr. Seth Grant made his record 5th appearance. I do this review every year because it gives us a chance to reflect on what we've learned together, and I enjoy tying together the ideas from different guests and their work. If you're new to brain science, it's not necessary to go back and listen to these episodes first. My goal is to give you an overview and hopefully motivate you to go back and enjoy the episodes you missed. I will also be scattering other useful information throughout the show. You'll be able to find all this in the show notes, so feel free to sit back and enjoy. We started the year with episode 165 with an encore interview of Stephen Macknik and Susana Martinez-Conde, neuroscientists who wrote the book Sleights of Mind, What the Neuroscience of Magic Reveals About Our Everyday Deceptions. Stephen and Susana studied the neuroscience of vision, but this book is about what they learned about how the brain works from working with several of the world's leading magicians. Susana emphasized that focused attention actually suppresses our attention to peripheral items. Magicians exploit this, they use misdirection and so we don't see their sleight of hand. But this brain behavior has implications for our decision making. That's why Susana recommended if we're making an important decision, we should make a list of each point, whether it's a logical reason or an intuitive one. But the key idea here is then take time to focus on each point separately for a few minutes before you make a decision. I also like the idea of sleeping on it, since that gives the brain a chance to process even more deeply. In episode 166, Stephen Macknik returned to tell us about his latest project, which involves trying to create a visual prosthesis that mimics the role of the fovea of the retina. This is a fairly technical episode, and so I've included in the show notes a link to a video that he made to make it a little easier to grasp. The project involves combining techniques from optogenetics and basic discoveries about vision. The key basic discovery is that each point on the retina has only 4 inputs: a right on, a right off, a left on, and a left off. Those 4 pieces of information categorize the quality of each point on the retina. Primates are the only mammal that has a fovea, which is the part on the retina that does detailed vision, which is one reason why research about vision is usually done on primates. Macknik said that the idea that all the low-hanging fruit regarding vision has been discovered is crazy because we have so much more to learn. He is really enthusiastic about his field and encourages more people to enter. Vision neuroscience combines both technology and basic science, and the discoveries are likely to be extended to other fields, such as making new kinds of prosthetics. In episode 167, I finally got to talk to French neuroscientist Stanislas Dehaene. He had a new book out this year called how we learn, why brains learn better than any machine for now. You may recall that in 2019, I talked about his book, Consciousness and the Brain, as a part of episode 160. So we briefly talked about consciousness, and he reminded us that most of the computation of the brain is unconscious. But our focus for this conversation was what happens in the brain when we learn. The goal is to be able to learn better. We also focused on how babies learn. Dehaene calls babies the world's best supercomputers. Babies are not blank slates. They start with innate abilities such as statistical learning and basic assumptions about physics. But their brains are also incredibly plastic. We talked about the difference between artificial intelligence and human learning. Current AI mimics, to a certain extent, the unconscious aspects of brain function. But humans need less data. There's a social component to our learning, and the theory of mind seems to be very important. We have the ability to abstract information. So I can give you the gist of a story. Dehaene says that we can synthesize the information in a way that we can talk about. We explored reading as an example of several principles. One is neuronal recycling, which means using parts of the brain that were originally devoted for something else. In the case of reading, that's using parts of the brain that were originally devoted to face and object recognition. Now, this means that we have to overcome some assumptions about so-called object invariance. That's the fact that when you look at somebody, it doesn't matter if you're looking at them from the left or from the right. So with object invariance, the little b and little d would be the same letter. So we have to learn that they aren't. This is one of the things that makes learning to read as an adult difficult. Reading does become automatic, but the decoding of the individual letters still goes on unconsciously. That's the reason why whole language or whole word approaches don't work during the learning stage. Dehaene emphasized the importance of using what science has discovered to improve how children are taught. We didn't discuss the problem within education of resisting scientific evidence in favor of tradition. The principles of learning do apply at all ages, and Dehaene likes to describe these as what he calls the 4 pillars: attention, active engagement, error feedback, and consolidation. Error feedback leads to a surprising discovery, which is that if you are trying to learn something new, you actually do better if you give yourself frequent self-tests instead of just constantly reviewing the material. Constantly reviewing the material gives you the false impression that you know more than you do. Testing is really important. I really encourage you to share this episode with parents and teachers. In episode 168, I talked with Cecelia Heyes about her book, Cognitive Gadgets: The Cultural Evolution of Thinking. Heyes is a professor at the University of Oxford who studies the way the human mind differs from that of other animals. This is one of my favorite episodes because she introduced us to the concept of cognitive gadgets, which was entirely new to me. It's always exciting to be able to share something new that expands our understanding of how our brain makes us human. I was very fortunate to interview Dr. Heyes shortly after talking with Stanislas Dehaene because these two interviews complement each other. A cognitive gadget is the cognitive ability that is learned from others. Reading is an example. A cognitive instinct is an innate ability. An example would be associative learning. Hayes' book focuses on 3 abilities that are presumed to be unique to humans: imitation, mind reading, and language. Many researchers consider all 3 to be cognitive instincts, which implies that they are inherited via genes rather than being learned from the culture around us. Dr. Hayes' own research has focused on imitation and mind reading. She and her team have compiled compelling evidence that both are actually learned. We discussed the specific example of imitation, which included a landmark study done in Brisbane that demonstrated that newborn babies do not have the ability to imitate, but they do learn it very early on. A key point to remember is that studies in child development, and especially of very young babies, are revolutionizing our understanding of the importance of learning early in life. Dehaene also emphasized that. The example of language is very important, so I want to emphasize that Heyes actually started with the assumption that language is a cognitive instinct. She spent 6 months reviewing the literature, applying the same criteria as she did for imitation and mind reading. To her surprise, she found that the early evidence that appeared to support Chomsky's claim that language must be innate did not stand up to the larger body of more recent evidence. One interesting point is that several genetic variations that were originally presumed to support Chomsky's theory have been shown to relate to more general learning abilities, such as the ability to learn sequences. Thus, our innate ability to learn language appears to rely on several factors. One is our improved ability to learn in general, such as learning sequences. Another is our improved working memory. But actually learning a language requires cultural learning, which makes it a cognitive gadget, not an instinct. Now, if you have a skeptical response to what I just said, I encourage you to read Heyes' book and to look up the references for yourself, because as Patricia Churchland said during her last interview, the data don't lie. Why does this matter? The goal of brain science is to bring you discoveries from neuroscience that contribute to our understanding of how our brain makes us human. Does it matter whether some or many of our cognitive skills are learned or culturally acquired? Obviously, Dr. Heyes and I would answer yes. She pointed out that this is both good news and bad news. The good news is that the abilities can be studied scientifically in living humans. We aren't forced to rely on speculations from fields like anthropology. But the bad news is that any skill that is culturally acquired is fragile. It could be lost in a disaster. This is a sobering thought, but let's focus on the positive. A better understanding of how these skills are acquired can help us to teach more effectively. A good example would be teaching people with autism how to read minds more effectively. I view the interviews of Dehaene and Heyes as being very complementary, and I highly recommend listening to them together. I want to take a moment to remind you to check out my favorite app, Text Expander. It allows you to get time back by saving repetitive typing and avoiding mistakes. You create snippets for things that you type over and over again, and this really saves a lot of time. It helps you stay consistent and accurate, and you can include anything from text to images in your snippets. Text Expander is available for Mac, Windows, Chrome, iPhone, and iPad. All you have to do is visit textexpander.com/podcast for 20% off your first year. That's textexpander.com/podcast. And don't forget to tell them that you heard about it on Brain Science. Episode 169 was the encore playing of the interview with R. Douglas Fields, author of The Other Brain, which is a book about the glial cells that was published back in 2011. Glial cells are the non-neuronal cells in the nervous system. They outnumber neurons by at least 2 to 1, possibly more. And for a long time, glial cells were presumed to act only as structural support cells. The key idea of this episode was that glial cells do a lot more than just offer support. We now know that they have a role in signaling, and this means that they're important in diseases and in brain plasticity. Since this interview was originally recorded in 2011, I asked Dr. Fields to review the transcript to give me updates, and he said that the material was still quite up to date, but he did add some information about a recent discovery that shows now that the signaling in axons can actually stimulate myelination. That means that there's a glial cell, the oligodendrocytes, that makes myelin. And so it appears that they actually sense the signal going through the axon and make more myelin. This is really cool, and it suggests that brain activity in the young is going to determine which axons are myelinated, because the more active neurons are going to be stimulating more myelination. Episode 170 features Andreas Nieder, a German neuroscientist who wrote the book A Brain for Numbers, The Biology of the Number Instinct. Back in episode 168, we learned the distinction between a cognitive instinct and a cognitive gadget. Our discussion with Andreas Nieder about numbers gave us the opportunity to see examples of both. First, we discussed the surprising discovery that many animals, even honeybees, have an innate approximate number system, or a number instinct. This follows the so-called Weber-Fechter law that was originally discovered in the context of perception. What this means is that as the number of objects increases, it's harder to detect differences. It's easier to tell the difference between 1 and 2 objects than it is to tell the difference between 9 and 10 objects. This nonlinear relationship is actually logarithmic, which is the Fechter part of the law. Sensory neurons show this same behavior, sound being a good example. The louder sounds are, the harder it is to tell small differences. It has been demonstrated that babies as young as 50 hours old can respond to the difference in the number of objects. And here was a fascinating discovery. If you ask a young preschool child to put numbers on a line, they will space them logarithmically. That means that they'll put the larger numbers closer together. But after they go to school, they'll space them out equally, which means that they've gone from an instinctive logarithmic view to a linear one. Symbolic math appears to be unique to humans, but it is based on this approximate number system that we share with other animals. We still don't know how the human brain changes when we learn symbolic math, but Nieder is looking forward to exploring that in the future. A few other points. People who live in cultures without symbolic math still demonstrate the approximate number system. Language and mathematics appear to be separate, independent systems. So brain damage can cause isolated damage to one or the other ability. In terms of cognitive instincts and cognitive gadgets that we explored with Cecelia Heyes, the approximate number system is a cognitive instinct, while the symbolic math system is a clear-cut cognitive gadget. The fact that our unique symbolic math abilities are based on the approximate number system could provide new ways of predicting who's going to struggle, and eventually could lead to finding new ways to help those who have deficits in the approximate number system. Because the inability to do basic math is just as disabling as not being able to read. Math and reading are separate skills, even if reading does contribute to the ability to learn symbolic math. I want to take just a moment to talk to you about how you can support brain science financially, since I hope this review is giving you a better sense of the time required to create this show, even though I now outsource the audio editing. There are currently 3 ways to support the show: Premium, Patreon, and direct donations via PayPal or Venmo. Premium and Patreon are both methods of supporting the show in an ongoing manner. The premium subscription gives you unlimited access to all episodes and transcripts, with content going back to 2006. Another advantage of premium is that you can access all this extra content via the free Brain Science mobile app or via your browser. You can pay monthly, every 6 months, every year, and you can cancel anytime. Patreon is a good choice if you want to customize your contribution to fit your budget, or if you're interested in ad-free content. You pick whatever monthly amount you want to pay, but the benefits are tiered. If you contribute at least $3 a month, you have access to new transcripts every month. If you're able to afford $10 a month, you get the transcript and an ad-free version of each new episode. along with occasional bonus content. I call the $10 a month tier the Brain Science Lovers, and recently members of this tier were also invited to my webinar, Embracing Uncertainty. Obviously, it would be helpful to have more brain science lovers, but the downside of Patreon compared to Premium is you can't access the content via the free Brain Science app. If you are interested in learning more about these options, including how to make a direct donation, please go to brainsciencepodcast.com/donations. In episode 171, I interviewed Matthew Cobb about his book, The Idea of the Brain. Dr. Cobb is a neuroscientist in the UK who he's interested in how the nervous system evolved, and he studies smell in flies. In fact, he has another book out in 2020 called Smell, A Very Short Introduction. He was actually my very first podcast guest ever on my other show, Books and Ideas, back in 2007. He's written a couple of other books about the history of science, one about reproduction and the other about the discovery of DNA. But in his new book, The Idea of the Brain, he explores how people's ideas about the brain have changed throughout history. In the 19th century, the key metaphor was the telegraph system. Then, of course, around the middle of the 20th century, it shifted to the computer. I don't know what it's going to be in the 21st, maybe the internet. At any rate, I think this gives us some insight into how science really works. It's more than an accumulation of facts because the metaphors directly affect the questions people ask. Now, one long-standing opposition in neuroscience has been localization versus distributed or network models. And of course, neuroscience and our understanding of the brain is influenced by discoveries in other fields. For example, electricity was a key discovery that changed the way we see the brain. Should remind you that neuroscience is a pretty young field. The Society for Neuroscience was founded in 1969. We talked a little bit about Darwin's role. He saw the brain as a genetically inherited structure, not a blank slate. Another interesting scientist from the 19th century is Hermann von Helmholtz. He was the first to propose the predictive functions of the brain, which we now talk a lot about. He also proposed the idea that the brain constructs the world we experience rather than providing an accurate window. He discovered the visual blind spot, and he argued that visual processing is unconscious. Now, nobody really paid much attention to his ideas at the time because they didn't fit the existing paradigms. Around 1950, appreciation of the chemical nature of the brain and the emergence of computer metaphors became prominent. We talked about Candace Pert's role in the discovery of opioid receptors. This was important to both of us because she is an unrecognized contributor to this field. Then, of course, we did talk a little bit about the connectome, kind of emphasizing its limitations, which is to say having a wiring diagram doesn't really tell us how the brain works. One of the things that stood out for me as I read The Idea of the Brain was how often key ideas have been discovered in the past but forgotten, either because they didn't fit the current paradigm or metaphor, or because the technology was not yet developed enough to answer the questions raised. Two things influence the questions we ask: our current paradigm and our current technology. One thing we didn't have time to talk about was what Cobb called neuromythology, which is when an idea becomes popular even when it has been discarded by mainstream neuroscience. An example of this would be the triune brain, which was already known to be wrong when Carl Sagan made it popular in the 1970s. Now, don't get me wrong, I loved Carl Sagan, but this was an example of the hazard of trying to keep up with science outside one's own field. The Idea of the Brain is an engrossing introduction to the history of neuroscience, and I highly recommend it, especially if you are new to the field. It would also make a great gift for any students who are interested in neuroscience and for anyone who's interested in the history of ideas. episode 172, I interviewed György Buzsáki, who first appeared back in episode 31 when we talked about his first book, The Rhythms of the Brain. This time we talked about his new book, The Brain from the Inside Out. Buzsáki is a pioneer of exploring the role of oscillations in brain function, and he's spent his career doing experiments in the hippocampus, the deep structure that is involved in memory and in the sense of place. The goal of this book was to challenge the paradigms that we've inherited from our giants, the psychological terms inherited from William James and the other founders of psychology. He argued that these terms don't necessarily correspond to what the brain actually does. A little while ago, I talked about the fact that neuroscience is a pretty young field. It's actually not it came after psychology, and it has followed psychology, asking the same question as psychology once asked about the mind. How does the brain reveal the world? There was this idea of perception leading to action, the so-called perception-action cycle. Buzsáki argued that our typical approach distorts our understanding because we know what the brain is doing and we have information about the outside world, so we can then make correlations. However, as Buzsáki said, the neurons in the visual cortex don't know anything about vision. He also said we inherited a frame that did not include the brain because psychology was built on terms such as emotion and perception. So again, there was that assumed sequence, perception going through the black box of the mind, leading to action. He calls this the outside-in approach because the goal is to find a correlation between the world and what the brain does. In contrast, the inside-out approach recognizes the importance of the spontaneous brain activity. He emphasized this key idea. It is not enough to show a correlation between brain activity and outside events. You must show that the pattern matters, which means it has to change the neurons downstream. The outside-in approach tends to see the brain as a passive receiver of information. Ironically, Buzsáki sees behaviorism as a valuable step because although the mind was seen as a black box, at least it wasn't seen as passive. Another key idea from the inside-out approach is that it challenges the blank slate view of the brain. Buzsáki shared several key axioms of his inside-out approach. 1, the brain is a self-organized system. 2, to understand perception, one must start with action. There's no need for sensors if you can't respond. And 3, it's never sufficient to just measure a correlation. You must demonstrate that it has an effect. A good example of when this isn't true is with EEGs. The brain doesn't appear to use EEG signals, even though we can measure them and make correlations. Buzsáki's key claim is that learning is the brain's matching of its spontaneous activity to outside events that coincide. When you take the spontaneous activity of the brain seriously, this leads to some surprising discoveries. The logarithmic behavior of sensory neurons makes sense because it allows them to respond over a larger range of signals. And also, some neurons are more plastic than others, which allows us to learn new things while perceiving knowledge. Buzsáki observed that neuroscience currently lacks a framework such as the standard theory of particle physics. His hope is that the inside-out approach will inspire new approaches and open up new questions. The Brain from the Inside Out is a challenging, thought-provoking book. It's full of experiments that demonstrate the ideas that we discussed. It's an important book for anyone considering a career in neuroscience, and it's likely to join his first book, Rhythms of the Brain, as a classic in the field. I'm going to skip 173 for a minute and talk about 174 because reading Buzsáki's The Brain from the Inside Out actually motivated me to go back and read Georg Northoff's book, The Spontaneous Brain, From the Mind-Body to the World-Brain Problem. That was actually published back in 2018 but I talked to him about it in episode 174. This fascinating book combines ideas from the philosophy of mind with growing evidence of the brain's spontaneous activity. Northoff's rather startling solution to the mind-body problem is to eliminate the term mind. By moving the focus to what he calls the world-brain problem, he claims that mental features like consciousness and self can be traced to the way the brain aligns with its environment. His book is full of experimental evidence, which we could only touch on during our conversation. But first, I need to review a few technical terms. It's important to distinguish between the brain's spontaneous activity and so-called stimulus-induced activity. Northoff sees spontaneous activity as the basis of subjectivity because we now know that the so-called default mode network correlates with thoughts about the self, and the default mode network is active when we're not doing other thoughts or activities. A key idea is that external stimuli are not the sole determinants of stimulus-induced activity. Stimulus-induced activity and the spontaneous activity interact in a nonlinear or non-additive way, but the spontaneous activity goes beyond prediction. The stimulus-induced activity represents only about 5% of the brain's activity. The rest is spontaneous. You may recall that Buzsáki made the same observation, and they both referred to the predictive coding theory of Carl Friston. If you want to know more about predictive theory, I suggest starting with episode 126, where I talked with Andy Clark about this. Northoff's definition of consciousness is the capacity to experience yourself in a perspectival way as part of the wider world. Northoff feels that his approach to consciousness, which he calls a temporal spatial approach, is different from global neuronal workspace theory because he sees that as a purely cognitive approach. He sees access consciousness, which is what Dehaene works on, as only a subset of consciousness. He also sees his approach as less content-oriented. He argues that by integrating the resting state, the pre-stimulus state, and task-evoked activity, one may create a template for understanding consciousness. This is a very dense episode. Rather than trying to summarize it further, I've tried to give you a sense of what it's about. I hope you'll go back and listen if you missed it. And if you share my fascination with consciousness, you may want to listen to it again. I see Buzsáki and Northoff's approaches as being very complementary since they both emphasize the importance of spontaneous brain activity, but they build their arguments on different sets of experimental data. Buzsáki proposes that the brain essentially learns to associate its spontaneous activity with outer events, while Northoff emphasizes the role spontaneous activity plays as an essential component of consciousness. They both emphasize that 90 to 95% of the brain's activity is spontaneous and that this has important consequences, such as contributing to its stability in facing a changing environment. This appreciation of spontaneous brain activity represents an extreme shift from seeing the brain as a passive receiver of inputs from the outside world. That's why Northoff sees this approach as analogous to the Copernican revolution. Just as the Earth is no longer the center of the universe, he argues that we should shift from the mind-brain problem to the world-brain problem. Ultimately, he argues that this is a problem that can be solved. For those of you who are new to brain science, I should mention that the discussion with Buzsáki and Northoff builds on the four-part discussion of consciousness that you can find in episodes 160 through 163. I want to remind you about our new show sponsor, the Neurology Podcast. The Neurology Podcast is the original neurology podcast since 2007 from the journal Neurology and the American Academy of Neurology. The American Academy of Neurology is the largest association of neurologists and neuroscience professionals. Listen to interviews with authors of the most exciting research articles on weekly podcasts. Subscribe to the Neurology Podcast wherever you get your podcasts, or visit aan.com/podcast for more information about the show. Episode 173 is a solo episode where I read an excerpt from the second edition of my book, Are You Sure? The Unconscious Origins of Certainty, which was released in June 2020. I want to thank everyone who bought this book over the last 6 months. For new listeners, I will mention that this book is based on several early episodes of Brain Science that featured the work of retired neurologist Robert Burton. Dr. Burton graciously allowed me to publish our conversation in book form. Are You Sure? Is available in both paperback and ebook. You can buy the ebook at brainsciencepodcast.com or from your favorite online bookseller, where you can also get the paperback. If you're interested in an autographed copy of the paperback for $25, send me an email at brainsciencepodcast@gmail.com. I'm still getting requests to release an audio version of Are You Sure? The main reason I haven't done this is that much of the book contains quotes from Dr. Burton, which means an audio version would require a second male narrator, making it too expensive to produce. However, I want to point out that you can still get the original episodes that form the core of the book, since it's based on episode 42, 43, and 96. If you're a premium subscriber, you already have access to these episodes, and you can literally buy all 3 at brainsciencepodcast.com for less than the price of the ebook. Finally, I want to remind you that the real goal of this book is to reach people who don't listen to Brain Science, so I hope you'll consider sharing your copy with someone else, and please consider posting a review on Amazon or Goodreads. Episode 175 features Carol Tavris, co-author of Mistakes Were Made, But Not By Me, the key book about cognitive dissonance. Not surprisingly, this episode elicited a strong emotional response from several listeners. In contrast to our conversation about the first edition of this book back in 2010, it was inevitable that the current political events came up during our conversation. But I want to emphasize that the book contains examples of the same kind of behavior on both the left and the right, because the point we were trying to make is that everyone experiences cognitive dissonance and that the natural response is rationalization. Even though cognitive dissonance is a psychological theory, it's important to appreciate that its predictions have been replicated in literally thousands of experiments since it was first proposed by Leon Festinger back in the 1950s. The basic idea is that if we're faced with a fact or action that contradicts our prior beliefs, we will automatically begin to rationalize to reduce the internal discomfort this produces. This allows us to live with our mistakes, but it can also blind us to the need to change our position or to admit that we were wrong. If you're only going to read one book from this year's list, it should be Mistakes Were Made, But Not By Me, Why We Justify Foolish Beliefs, Bad Decisions, and Hurtful Acts by Carol Tavris and Elliot Aronson. Next, we come to episode 176, where Dr. Seth Grant made his record 5th appearance. We've gotten into the habit of talking every couple of years so that he can tell us about his latest research. But this year, I asked him to start out by giving us an overview of his amazing career. Dr. Grant's training was in molecular biology, and as a postdoc, he helped bring the use of transgenic mice to the lab of Nobel Prize winner Eric Kandel. This was an important step in bringing the tools of molecular biology to neuroscience. The first time I talked with Grant back in 2008, he had recently made a very surprising discovery. He had set out to determine the protein makeup of the post-excitatory synapse, and he had discovered that it was much more complex than previously imagined. Eventually, he realized that a single synapse actually has 500,000 to 1 million different proteins, and that these proteins are assembled into protein machines that he calls super sophisticated molecular computers. He has also explored how several of these structures are involved in behavior such as learning. Another surprise was that the synapse complexity actually evolved before complex brains and nervous systems. Combining genetic discoveries and techniques to uncover the protein structure of the synapse has led to more fascinating discoveries, such as the fact that mutations known to be associated with schizophrenia can be traced to specific proteins in the synapse. On an earlier episode, we talked about an experiment that showed that these same mutations cause learning deficits in mice. But some of this molecular machinery actually appears in unicellular organisms, which means that the molecular machinery for behavior goes back to unicellular organisms. Prior to this research, it was assumed that all synapses were more or less the same, but Grant's team has shown that the proteins in the vertebrate synapse are more complex than that of invertebrates. Grant reminded us that this was made possible by two duplications of the entire genome that occurred during the Cambrian explosion about 500 million years ago. This means that after the second duplication, organisms had 4 copies of each gene. This allowed a lot more genetic variation to occur while preserving the genes that were critical to survival. This double gene duplication was probably the event that made the Cambrian explosion possible. The bottom line is that the genomes of the vertebrates became more complex. This allowed greater complexity of molecular processes, bodies, and behavior. I should emphasize that this discovery was not made by Grant or his colleagues, but it has greatly influenced the direction of his subsequent work. Once Grant realized that the protein structure of the synapse is more complex than expected, he went on to show that this structure is not the same for every synapse. Not only are vertebrate synapses more complex than those of invertebrates, but within a single brain, different synapses contain different proteins. Thus, he moved from a focus on the proteome, which is characterizing the proteins of the synapse, to an attempt to map what he called the synaptome, which means mapping the distributions of the kinds of synapses across the brain using a mouse. In 2018, he released the first synapse of the mouse brain, and he showed that the synaptome changes predictably during the life of the mouse. He has developed a technique for tagging 2 of the most important complexes so that their distribution across the brain can be described. His latest paper shows that these particular proteins seem more prominent in parts of the brain associated with cognition, the cortex and the hippocampus. I should note that this discovery parallels work by others that has shown that gene expression in the brain varies throughout the brain and changes during the lifespan. I can't overemphasize the importance of this discovery. Equally important, Grant and his colleagues are sharing their data and techniques with the scientific community. Grant always emphasizes how his work is built on that of others. For example, the technique of using fluorescent labels of different colors allowed his lab to identify which synapses contain which proteins. I want to point out that as a molecular biologist, Grant brings tools to his work that I suspect have helped him make unique contributions to neuroscience. Modern neuroscience requires an interdisciplinary approach that incorporates the tools and discoveries from a wide variety of fields. That's why I encourage students not to narrow down your studies prematurely. Learn as much as you can from other fields because you never know when this could lead to something big. Grant made one other important point during this year's interview. He pointed out that synaptic diversity implies the ability to store much more information in the synapse because different synapses are likely to respond slightly different to the same stimulus. He observed that this might even provide an understanding for how memories are encoded. He also pointed out that this discovery could suggest new approaches to designing artificial intelligence, since current models assume identical synapses. I realize that this has been a long discussion, but because I know these discoveries are still not widely known, I want to emphasize their importance by summarizing my summary. So here are the key ideas. Synapse complexity happened before the evolution of brains and complex nervous systems. The complexity of vertebrates was made possible by the double gene duplication that occurred during the Cambrian explosion. Gene expression in the brain appears to change predictably during the lifespan. Synapse complexity varies by brain area. Parts of the brain involved in so-called higher function are more complex. Genetic mutations associated with problems like schizophrenia appear to impact synapse function. Okay, now let's narrow it down to the three most important ideas. 1, the synapse is more complex than anyone expected. 2, this complexity began before brains and complex nervous systems. And last, within a given brain, synapses are diverse and the complexity mirrors function. A few months ago, I announced my intention to create an edited compilation of the transcripts of all 5 of Dr. Grant's interviews. And at the time, I also asked for feedback about other possible compilations. To be honest, there wasn't much of a response, and the project is stalled. I probably won't pursue it further unless I get more feedback, indicating significant interest in the project. But at this point, I want to give a shout out to Diane Jacobs, who was an earlier supporter of the show and who created the first episode transcripts. Diane was also the person who alerted me to the work of both Seth Grant and György Buzsáki. Thank you, Diane, for helping make this a much stronger show. I want to take just a moment to talk about transcripts in general for the sake of new listeners. Transcripts exist for every episode of Brain Science dating back to December 2006. The transcript for today's episode and all previous review episodes are free. You will find links in the show notes, and you can also access these via the free Brain Science app. You can also buy individual episode transcripts at brainsciencepodcast.com, but you can get unlimited access to all the transcripts via the premium subscription. Okay, we're heading down the home stretch. 2 more episodes to go. Episode 177 features Bernard Baars and his friend David Edelman. I invited Dr. Baars onto Brain Science because his global workspace theory inspired many of the current theories of consciousness. When he first proposed it back in 1980, consciousness was still considered taboo. It was fascinating to hear him talk about how behaviorism stymied the scientific study of consciousness and how the so-called cognitive revolution allowed scientists to break free and allowed psychology to return to its roots to study the mind and consciousness. The easiest way to think of global workspace theory is to see it as the psychological theory that inspired global neuronal workspace theory. But any theory that includes some sort of widespread signaling process is probably compatible with Baars' original idea. Last month's episode, 178, which was an interview with Peter Sterling, author of What is Health, Allostasis, and the Evolution of Human Design, had some technical difficulties. So before I summarize the key ideas, I want to emphasize that I do not plan to go to shorter episodes. My goal is to continue to make the episodes about an hour long. The key idea of episode 179 was to highlight the predictive or allostatic behavior of the brain. We've talked about this predictive behavior many times because it's an important theme in contemporary neuroscience. But Sterling's unique contribution is to point out the implications for our understanding of health and disease. He pointed out that medical students are taught about homeostasis, but this is really the secondary corrective mechanism that is triggered when the brain's predictions fail. Ideally, the prediction errors will be very small. For example, under normal conditions, small surprises cause small surges of dopamine, which we perceive as highly motivating. And stress leads to a temporary surge of cortisol. Unfortunately, we now have the ability to create abnormally large dopamine surges through the use of drugs and other extreme conditions. And this causes the receptors to recalibrate so that we're no longer satisfied with the small dopamine surges that have functioned well for thousands of years. Similarly, chronic stress leads to constant elevation of cortisol and is linked to many of the diseases of so-called modern life. Dr. Sterling's book also pointed out that the disadvantaged populations are especially affected by these trends. I was particularly struck by the idea of the importance of the small dopamine surges. I found myself wondering if we can recalibrate our dopamine receptors by making an intentional effort to generate these physiological surges by practices like gratitude. Unfortunately, we have the flip side of the increasing use of drugs and alcohol being triggered by the social isolation of COVID-19. That leads me directly to reminding you how important your feedback is to me. Your emails give me a healthy dopamine surge, which in turn motivates me to keep creating the show. I'd love to hear how you're coping with the current situation, or if that feels too personal, just tell me how you found the show and how long you've been listening. Do you have a favorite book you think I should feature in 2021? Don't forget that even if you can't afford to support Brain Science financially, you can share the show with others, which helps Brain Science grow. Before I close, I want to remind you that you can find complete show notes and episode transcripts at brainsciencepodcast.com. Brain Science is produced independently and relies on the financial support of listeners like you. To learn more, please visit brainsciencepodcast.com/donations. And of course, don't forget to send me feedback at brainsciencepodcast@gmail.com and post to our Brain Science Podcast fan page on Facebook. If you'd like to get the free newsletter and get show notes automatically, you can text Brain Science, all one word, to 55444. That's Brain Science, all one word, to 55444. Thanks again for listening. I look forward to sharing more neuroscience with you in 2021. Brain Science is copyright 2020 to Virginia Campbell, MD. You can share this audio with others, but for any other uses or derivatives, please email me at brainsciencepodcast@gmail.com. The theme music for Brain Science is Mindfire, written and performed by Tony Catraccia. You can find his work at syncopationnow.com.